Today, we're delving into the intricate world of the nervous system, specifically focusing on how neurons generate and process signals. Our material for this discussion comes from Professor Vanessa's lecture presentation on the resting membrane potential, action potential, and information processing. Let's start with the resting membrane potential, which is the electrical potential difference across the cell membrane of an unstimulated resting cell. You see, all neural activity fundamentally begins with a change in this resting membrane potential. The extracellular fluid, or ECF, and the cytosol, or intracellular fluid, have very different concentrations of ions. The extracellular fluid is rich in sodium, Na+, and chloride ions, while the cytosol primarily contains high concentrations of potassium ions, K+, and negatively charged proteins. What's crucial here is that membranes are selectively permeable, meaning they control what passes through. At rest, ions move passively through what we call leak channels, which are always open, but they also move across the membrane via active transport, most notably through the sodium-potassium exchange pump. Membrane permeability varies by ion. For instance, more potassium ions leak out of the cell than sodium ions leak into it. And those large negatively charged proteins inside the cell, they're simply too big to cross the membrane surface. This combination of factors means that the inner surface of the membrane is more negative compared to the outside when the cell is at its resting membrane potential.
Now, let's explore the electrochemical gradient. When we talk about current, we're referring to the movement of charges to eliminate a potential difference. Resistance, on the other hand, measures how much the membrane restricts ion movement. If resistance is high, the current is small, and vice versa. Importantly, this resistance changes when ion channels in the membrane open and close, allowing ions to move across the membrane driven by passive processes, which are the gradients. There are two main components to this gradient. First, the chemical gradient. This is the passive movement of ions driven by their concentration gradient. Driven by their chemical gradient, sodium ions move into the cell and potassium ions move out of the cell. Second, the electrical gradient. This is the passive movement of ions due to differences in charge. Driven by the electrical gradient, sodium ions move into the cell because of the negative charge inside, and surprisingly, potassium ions also move into the cell, drawn by that same negative charge. The electrochemical gradient is the sum of both these chemical and electrical forces acting on an ion across the membrane. At a neuron's resting membrane potential, the chemical gradient for sodium and potassium ions is stronger than the electrical gradient. For potassium, the chemical and electrical gradients are opposed, meaning the net electrochemical gradient tends to force potassium ions out of the cell. For sodium ions, the chemical and electrical gradients are combined, forcing sodium ions into the cell. This electrochemical gradient represents a form of potential energy, ready to be utilized.
We also have something called the equilibrium potential, which is the membrane potential at which there is no net movement of a particular ion across the cell membrane. If the plasma membrane were freely permeable to potassium ions, the outflow of potassium would continue until the equilibrium potential was reached, which is quite close to the resting membrane potential. For sodium ions, however, if the membrane were freely permeable to sodium, the inflow would continue until its own equilibrium potential was reached, which is very different from the resting membrane potential. To counteract these passive movements and maintain the resting membrane potential, active transport is essential. The sodium-potassium exchange pump is a prime example. This pump is powered by ATP and actively moves 3 sodium ions out of the cell for every 2 potassium ions it brings into the cell per ATP.
It's constantly working, opposing the passive leaking of sodium in and potassium out through leak channels, driven by their electrochemical gradients, thus keeping the resting membrane potential stable.
Changes in the resting membrane potential result from opening or closing of specific membrane channels in response to stimuli. These are known as gated ion channels, and they are typically closed when the cell is at rest. We classify them into three main types. First, there are chemically gated or ligand-gated ion channels. These open only when they bind to a specific chemical or ligand like acetylcholine, ACH. You'll primarily find these on the cell body and dendrites of neurons. Second, we have voltage-gated ion channels. These open and close in response to changes in the membrane potential itself. They're critical for nerve impulses and are found in the axons of neurons and the sarcolemma of skeletal and cardiac muscle cells. Voltage-gated sodium channels are particularly interesting because they have two gates, an activation gate and an inactivation gate, allowing them to exist in three states: closed but capable of opening, open or activated, or closed and incapable of opening or inactivated. And third, mechanically gated ion channels. These open and close in response to physical distortion of the membrane. They are typically found in sensory receptors that respond to touch, pressure, or vibration. Now let's discuss graded potentials, also called local potentials. These are temporary, localized changes in the resting membrane potential. They're produced by any stimulus that opens gated channels, and they spread passively outward from the point of stimulation, decreasing in strength with distance. Essentially, stronger stimuli produce stronger graded potentials.
Graded potentials can cause depolarization, a shift from the resting membrane potential toward a less negative potential. This happens when positively charged sodium ions enter the cell through gated channels. Depolarization can produce local current, the movement of ions parallel to the membrane surfaces, allowing the depolarization to spread. Conversely, graded potentials can also cause hyperpolarization, a shift toward a more negative potential due to positively charged potassium ions exiting the cell through gated channels. Repolarization is the process of returning to the normal resting membrane potential after depolarization, driven by the loss of positively charged potassium ions through gated channels and the action of the sodium-potassium exchange pump. Graded potentials aren't exclusive to neurons and muscle cells. They occur in many types of cells and often trigger specific cell functions, such as the exocytosis of glandular secretions. A classic example is at the neuromuscular junction, where ACH causes a graded potential at the motor end plate, which can then trigger an action potential in the muscle sarcolemma.
This brings us to the action potential, often called a nerve impulse. An action potential is a large depolarization that, once initiated, affects the entire excitable membrane and doesn't diminish as it moves away from its point of stimulation. It's stimulated by a graded potential that depolarizes the axolemma to threshold, typically between -60 and -55 millivolts. The all or none principle applies here. An action potential is either triggered or it isn't. Any depolarization that changes the membrane potential to threshold will cause an action potential that propagates all the way down the axon. If the threshold isn't reached, no action potential is generated. Furthermore, all action potentials are essentially identical in amplitude, no matter how large the stimulus, as long as it met the threshold. Let's break down the generation of an action potential into steps. Step 1 is depolarization to threshold, caused by a graded potential large enough to depolarize the plasma membrane of the initial segment to threshold. Step 2 is rapid depolarization. Voltage-gated sodium channels open, activate, and sodium rushes in, driven by its electrochemical gradient. Step 3 is repolarization.
At about plus 30 millivolts, plus 30 MV, the voltage-gated sodium channels inactivate and the voltage-gated potassium channels open. Potassium ions then move out of the cytosol, causing repolarization. Step 4 is hyperpolarization. The voltage-gated potassium channels begin to close as the membrane reaches the normal resting potential. However, they are slow to close, so additional potassium leaves the cytosol, resulting in a brief hyperpolarization. After all voltage-gated potassium channels finish closing, the sodium-potassium exchange pump works to restore the resting membrane potential.
During and immediately after an action potential, there's a refractory period, where the membrane will not respond normally to additional depolarizing stimuli. The absolute refractory period is when the membrane cannot respond at all because the sodium voltage-gated channels are open or inactivated and unable to open. The relative refractory period follows. The sodium voltage-gated channels return to their resting state, but since the membrane is hyperpolarized at first, a larger than normal stimulus is needed to reach threshold. This period ends when the membrane returns to the resting membrane potential. Propagation is the series of steps that move the action potential along the axon. The entire sequence, depolarization, repolarization, and hyperpolarization, is repeated over and over down the axon. There are two main types of propagation. Continuous propagation occurs in unmyelinated axons. Step 1. An action potential develops at the initial segment and depolarizes the membrane to plus 30 millivolts. Step 2, a local current develops and depolarizes the next segment of the axon to threshold. Step 3, an action potential occurs in the second segment as the initial segment begins repolarization. Step 4, local current depolarizes the next segment to threshold and the cycle is repeated. The action potential travels down the axon because the local potential cannot depolarize segments that are in the refractory period. Saltatory propagation, on the other hand, occurs in myelinated axons. This method is faster than continuous propagation and requires less energy. The local current effectively jumps from node to node because only the nodes of Ranvier have the voltage-gated channels capable of firing an action potential.
Propagation speed depends on a couple of factors. Myelin increases propagation speed, and a larger axon diameter also increases propagation speed. Axons are classified into types based on their diameter, myelination, and propagation speed. Type A fibers, Type B fibers, and Type C fibers. Type A fibers are myelinated with a large diameter and transmit information to and from the CNS rapidly. Examples include fibers that carry sensory information, such as position and balance, and those that carry motor impulses to skeletal muscles. Type B fibers are myelinated with a medium diameter, transmitting information at intermediate speeds. Type C fibers are unmyelinated and have a small diameter, transmitting information slowly. Both Type B and Type C fibers carry most sensory information from the skin and carry motor instructions to smooth muscle, cardiac muscle, and glands.
Next, let's explore synapses, the specialized sites where a neuron communicates with another cell. The presynaptic cell is the neuron which sends the message, while the postsynaptic neuron or cell is the one that receives the message. There are two main types of synapses. Electrical synapses transmit signals via direct physical contact between cells. The presynaptic and postsynaptic membranes are locked together at gap junctions, allowing ions to pass between cells directly, which means the local current affects both cells and action potentials are propagated quickly. You find these in some areas of the brain, the eye, and ciliary ganglia. Chemical synapses, which are the most common type between neurons, transmit signals across a gap, called the synaptic cleft, using a chemical signal known as a neurotransmitter. These can be categorized based on where they occur: axoaxonic synapses between two axons, axosomatic synapses between an axon and a cell body, axodendritic synapses between an axon and a dendrite, neuromuscular junctions between an axon and a muscle cell, or neuroglandular junctions between an axon and a secretory cell. The function of chemical synapses involves neurotransmitters, which are chemical messengers contained within synaptic vesicles in the axon terminal of the presynaptic cell. Upon the arrival of an action potential, the neurotransmitter is released into the synaptic cleft by exocytosis. The neurotransmitter binds to receptors on the postsynaptic plasma membrane, producing localized changes in permeability and graded potentials. Whether an action potential is generated in the postsynaptic cell depends on the amount of neurotransmitter released and the sensitivity of the postsynaptic cell.
Cholinergic synapses are those that release acetylcholine. You find these at all neuromuscular junctions involving skeletal muscle fibers, many synapses in the CNS, all neuron-to-neuron synapses in the PNS, and all neuromuscular and neuroglandular junctions in the parasympathetic division of the ANS. Here's what happens at a cholinergic synapse. First, an action potential arrives at the axon terminal and depolarizes its membrane. Second, extracellular calcium ions enter the axon terminal and trigger exocytosis of acetylcholine into the synaptic cleft. Third, ACH binds to receptors, chemically gated cation channels on the postsynaptic membrane, and this depolarizes due to sodium ion influx, creating a graded potential. Finally, ACH is broken down by the enzyme acetylcholinesterase, ACHE, and its byproducts are removed from the synaptic cleft, ensuring the signal is brief and precise. There's also a synaptic delay, which is a delay between the arrival of the action potential at the axon terminal and the effect on the postsynaptic membrane. This delay is mostly due to the time required for calcium ion influx and neurotransmitter release. Fewer synapses lead to faster responses. Some reflexes involve only one synapse. Another phenomenon is synaptic fatigue. This occurs when the response of the synapse weakens due to insufficient neurotransmitter until the neurotransmitter has been replenished, typically when neurotransmitters cannot be recycled or synthesized fast enough to meet the demands of intense stimuli.
Moving on to neurotransmitters and neuromodulators. We classify neurotransmitters based on their effect. Excitatory neurotransmitters cause depolarization of the postsynaptic membranes, making it more likely for an action potential to be generated. Inhibitory neurotransmitters cause hyperpolarization, suppressing action potential generation. Crucially, the effect of a neurotransmitter on the postsynaptic membrane depends on the properties of the receptor it binds to, not on the nature of the neurotransmitter itself. Let's look at some specific classes. Acetylcholine, as we mentioned, is an excitatory neurotransmitter found in both the CNS and PNS, at NMJ and other synapses. Biogenic amines include norepinephrine, or NE, or noradrenaline, which is released at adrenergic synapses, has an excitatory and depolarizing effect, and is widely distributed in the brain and portions of the ANS. Under biogenic amines, we have dopamine, which may be excitatory or inhibitory and is used in many areas of the brain. Dopamine-producing neurons are damaged in Parkinson's disease, and cocaine inhibits the removal of dopamine from the synaptic cleft. Serotonin is a CNS neurotransmitter, and serotonin deficiency leads to depression. Antidepressant drugs of the SSRI, selective serotonin reuptake inhibitors, class inhibit the reuptake of serotonin by axon terminals, therefore making serotonin concentration in the cleft higher. Amino acids also function as neurotransmitters. Glutamate is the most important excitatory neurotransmitter in the brain, important for memory and learning. Glycine has an inhibitory effect in the CNS. The poison strychnine blocks glycine receptors. Gamma-aminobutyric acid, or GABA, also has an inhibitory effect in the CNS, and anti-anxiety drugs enhance the effect of GABA. Neuropeptides are small peptide chains that can serve as neurotransmitters. Substance P, for example, is a neurotransmitter at synapses that relay pain sensation and is also found in the digestive tract. Neuropeptides can also act as neuromodulators, chemicals that change the cell's response to a neurotransmitter by binding to receptors on presynaptic or postsynaptic membranes. Opioids relieve pain and bind to the same receptors as opium and morphine, including classes found in the CNS like enkephalins, endorphins, and dynorphins. Finally, dissolved gases can also act as neurotransmitters. Nitric oxide, NO, is used at synapses that innervate smooth muscle and in the brain. Carbon monoxide, CO, is also used in the brain.
Neurotransmitters and neuromodulators exert their effects via receptors in two main ways. First, through an ionotropic effect, which is a direct effect on the membrane potential by opening or closing chemically gated ion channels. Examples include ACH, glutamate, and aspartate. Second, through metabotropic effects. This is an indirect effect by binding to G protein-coupled receptors, where G proteins activate enzymes such as adenylate cyclase, which then activate second messengers such as cyclic AMP, as seen with NE, dopamine, serotonin, and GABA. Or it can involve an indirect effect by entering the cell and activating intracellular enzymes, as lipid-soluble gases like nitric oxide and carbon monoxide do.
Let's conclude by looking at information processing in the nervous tissue. A postsynaptic cell can receive thousands of incoming signals. The postsynaptic cell integrates incoming excitatory and inhibitory stimuli. The net effect of all incoming signals on the axon hillock ultimately determines if an action potential is produced. Postsynaptic potentials are graded potentials developed in a postsynaptic cell in response to neurotransmitters. Excitatory postsynaptic potentials are graded depolarizations that make it more likely for the neuron to produce an action potential. Inhibitory postsynaptic potentials are graded hyperpolarizations that inhibit the neuron from producing an action potential because its membrane potential moves further away from threshold. Summation integrates the effect of all incoming graded potentials. Temporal summation occurs when rapid, repeated stimuli arrive at a single synapse and their effects add up. Spatial summation happens when simultaneous stimuli arrive at multiple different synapses and their combined effects are integrated. Facilitation is bringing the membrane potential of the initial segment closer to threshold, induced by incoming EPSPs. The larger the degree of facilitation, the smaller the stimulus needed to trigger an action potential. The summation and interactions between EPSPs and IPSPs determine the membrane potential at the initial segment and the likelihood of firing an action potential. Neuromodulators and hormones can also change the membrane sensitivity to neurotransmitters. We also observe presynaptic regulation at axo-axonic synapses. Presynaptic inhibition decreases the rate of neurotransmitter release at the presynaptic membrane, while presynaptic facilitation increases it. Finally, the rate of action potential generation is proportional to the frequency of stimulation. If the membrane potential of the initial segment remains above threshold, more action potentials are produced. More depolarization above threshold results in a higher frequency of action potentials. A maximum rate of action potentials is reached when the relative refractory period is eliminated because there is a large enough depolarizing stimulus.
To summarize, information is relayed in the form of action potentials. Neurotransmitters released at a synapse may have excitatory or inhibitory effects, and neuromodulators can alter the rate of neurotransmitter release or the response of the postsynaptic neuron. Neurons may also be facilitated or inhibited by chemicals other than neurotransmitters or neuromodulators.
The response of a postsynaptic neuron can be altered by neuromodulators or other chemicals that cause facilitation or inhibition, by activity underway at other synapses, and by modification of the rate of neurotransmitter release through facilitation or inhibition. This detailed look at membrane potentials, action potentials, and how neurons process information, as presented by Professor Vanessa, highlights the complex electrical and chemical dynamics that underpin all neural function. If you found this explanation helpful, consider sharing it with someone curious about how our nervous system truly operates.